Hi, I'm Ellie. I'm a PhD student at SAMS and I'm researching biorefinery of microalgae with Michelle Stanley, Mikey Ross, Sebastian Jubo and Valeria Montelesco, and this is in collaboration with a company called Xanthella. So I'm studying biorefinery of microalgae. I'm working on two species, one called Chromachlorus fingiensis and one called Porphyridium or Eugenium. So basically what this involves is growing microalgae and then breaking the cells and then separating out the different products. The thing that people quite often don't understand is the fact you can actually get different products from the algae. Algae mainly made up of like proteins, lipids, carbohydrates, but within those you have so many different products, like such a wide array. So for example, proteins can be in the form of pigments or enzymes which can have completely different applications. You can use them for pharmaceuticals, cosmetics, food dyes, anything like that. So I guess it's difficult for people to grasp how broad the array of products is, and also in terms of biorefinery it's quite difficult to understand how you separate the products. So all the different products have different properties. So they could be soluble, insoluble, hydrophilic, hydrophobic, and the way they react to different methods of downstream processing will be different based on their structure. Chromachlorus fingiensis produces astaxanthin, which is a really valuable carotenoid, as well as other secondary carotenoids, which have high value. And a distinguishing feature of that, it simultaneously produces lipids, specifically tags, which can be used as biofuels as well as nutraceuticals. The Porphyridium species produces phycocyanin, which is a blue pigment that's also very valuable, and it also produces EPS, which is exopolysaccharides, starch, and proteins and lipids as well. So the two species I'm working on haven't really been researched in biorefinery before. The research isn't there yet. People have looked into the different steps in the process but haven't put them all together to create a sequential process. So that's what I'm aiming to do.
Hi, I'm Kelly Jones and I'm studying blue carbon in Scottish marl beds, and we're working with Nick Minos at University of Glasgow and also Heidi Burdett at Heriot-Watt University, and I'm also funded by Nature Scot and Marine Scotland. You will have all seen in the news that Scotland's trying to be net zero by 2045, and net zero means that you don't emit any CO2, you're just kind of balancing what you emit with what you absorb. We're looking at how much CO2 is absorbed by this type of red algae in Scotland. So the whole marl beds, they're a type of coralline algae, so they look like coral, but in fact they're seaweeds. We're answering a question about a habitat that's really understudied for blue carbon research, and it's only been up until recently that we know that they actually store carbon in the first place. So a lot of the research so far has been done in one bed on the west coast of Scotland, and my research is studying almost the whole of Scotland. We've got multiple sites, so we're really kind of exploding how much knowledge we have about blue carbon storage in marl beds. Blue carbon research has only really been around for the last decade, and we've started to understand some really crucial questions, but there's still an awful lot that needs to be answered. So one of the things that does need to be answered is what happens to organic material when it lands on sediment. There's not yet a very good metric to measure like how much of that carbon is actually buried in the sediment, and that's quite a lot of the research that we're starting to work on.
Hi, my name is SK. I'm a PhD student at the University of Edinburgh. My supervisors here are Andrea Semiyao and Margaret Graham, and I also have a supervisor at SAMS in Oban called Michael Ross. I'm currently researching how Cladophora species can be used for sustainable wastewater treatment and the removal of potentially toxic elements. What people don't often understand about my project is actually how algae can contribute to wastewater treatment. Algae, like plants and animals and people, they need certain nutrients, they need certain elements to survive. And also algae are often adapted to a resource-restricted environment. When they're exposed to some of these very high concentrations of things that they like to have, they can absorb them very quickly. In the case of polluting elements, this can be a really good thing. This means that algae are passively improving water quality in their area, and at the same time, they can also be absorbing CO2 during photosynthesis, meeting the needs of the wastewater treatment system, as well as removing carbon dioxide during the process. What I find really exciting about my project is that I'm using live algae rather than dried. Oftentimes in wastewater treatment scenarios, they tend to use dried algae. Using live algae means that you have the opportunity to use both passive and active metal uptake routes. In the case of dried algae, metals are only able to adhere to the surface of the algae. When you work with live sorbents, you're able to get active uptake of the metals. So this means that there is metabolism-dependent transport across the cell wall into the algae, where further detoxification takes place.